Hello, everyone. Thank you for the organizing committee for the kind invitation to share our work, and especially to Patricia and Obi. I really wish that this was in person, and I hope that the next meeting will not be virtual, but more of an in-person meeting. I have no disclosure except to say that when we uncovered the histone mutation I'm going to be telling you about, I thought that we had moved from the Ice Age to barely the Stone Age, but now with the lot of bioinformatic data that we're collecting, the beautiful models that we're able now to better generate, and our improved understanding of the mechanism of oncogenicity. I think that we are moving fast forward, almost like a Trekkie at the speed of light. And I hope you'll share this excitement based on what I'm going to be telling you about. Our story started in 2012 when we uncovered, jointly with a group of Susie Baker at St. Jude, the recurrent hotspots mutation in histone genes. Nobody thought that histone genes and histone protein would be ever mutated because those are amazingly conserved at the protein level throughout evolution. And this discovery in high-grade glioma of those recurrent histone mutation was considered recently as a milestone, as one of the milestones in cancer in the last 10 to 20 years. Why is that? Because it's a new mechanism of producing a tumor. And we're deciphering the Rosette Stone in order to be able to better understand and better treat those children that have intractable cancers when they have those mutations. And again, the other beauty is that those mutations are also beautiful tools to actually taste development and specific time point in development and how the epigenome is done and made of during those developmental time points. So what is it that we've discovered? In collaboration with a group of Stefan Pfister and Jacek Majewski, we've uncovered recurrent hotspot somatic mutation heterozygous that drive an amino acid substitution, either lysine 27 to methionine and very rarely to an isoleucine in any of the histone 3 variant, or glycine to an arginine and more rarely to a valine substitution in exquisitely the non-canonical histone 3.3. Those mutations are mutually exclusive to each other and mutually exclusive to the IDH mutation that were previously uncovered in glioma. And similar to IDH mutation, they need what I call partners in crime, and those are the same than IDH. The vast majority will have T53 mutation, and some will really need ATRX, and we have yet to understand the need for ATRX. And for the sake of time, I won't go further into those. But just to say that in glioma, those mutations are never found on their own, even if though they are clonal on the tumor. They need partnership mutations in order to express their oncogenic potential. The other striking finding that we had is that those mutations have a very specific tempospatial pattern. They are actually exquisitely associated with brain development. Why is that? If you have a child that has a glioma, and high-grade glioma is going to be in the brain pons, that's going to be labeled diffuse intrinsic pontine glioma. If this child is aged 3 to 5 years of age, the mutation that's going to be there are K27M in histone 3.1 or 2, associated mainly with ACVR1, it's a kinase, somatic mutation. A little bit older, still in the brain pons, 5 to 7 years old, is going to be K27M in histone 3.3, and this time associated with anything that can affect TP53 pathway. Slightly older, 7 to 12, and at another stage in younger adult, you have histone 3.3 K27M, mainly associated with TP53 alteration, and in about 10% of cases, either with point mutation that are somatic in FGFR1, or fusion or mutation or truncation that lead to activation in EGFR. And the rare cases in the brain midline where you don't have K27M, you have overexpression of a gene by the name of E-Zhip. And E-Zhip is EZH inhibiting protein and contains a sequence, as I will tell you later, that is a K27M, which led us to actually suggest that your brain midline, the diffuse midline glioma, are a story of a lysine 27 that's badly written, either through a K27M, and you'll see that, or through E-Zhip overexpression. If you're a little bit older, you go to the cortex. 13 to 35 years of age, temporoparietal cortex, you'll have a glycine 34 to arginine or a valine mutation in histone 3.3, always associated with TP53 and with ATRX mutation. A little bit older, and this time it's going to be the frontal lobe mainly. You'll have IDH mutation that's been described associated with TP53. And as an in-between, you'll have truncating mutation in SETD2, and SETD2 is an enzyme that leads to the trimethylation of lysine 36 this time, that are mutually exclusive with the glycine 34 mutation, which lead us to suggest that actually the hemisphere is a problem of a lysine 36 that's badly written because SETD2 is absent, or read because glycine 34 written because glycine 34 is impacting on the lysine 36 that's downstream, and IDH mutagenesis affect the lysine demethylases that monitor K27, lysine 27, and lysine 36. So I hope I'm convincing you based on this distribution in the brain and with age that pediatric and young adult gliomas are a story of a 2 lysine on a histone 3 tail that are badly written or read and that are developmental problems. So how is E-Zhip working? E-Zhip works exactly like K27M. It has actually, this gene is not at all conserved in mammals, in placental mammals. This is where it's present. And within this gene, there are 15 amino acids that are exquisitely conserved. And those amino acids have a K27M sequence that make us say that E-Zhip is actually a non-cohistone mimic. This work, the initial discovery of E-Zhip was through David Allison and Marcel Kuhl's work in posterior fossa group A ependymoma. And it's interesting because those group A ependymoma, when they don't have E-Zhip overexpression, they have K27M mutation. And the yin-yang is in the high-grade glioma. When you don't have K27M, you have E-Zhip overexpression. Those are close cousins, and they live in the brain midline and in the posterior fossa, which also is another argument to say that the lysine 27 there is quite an important mark during development. And actually, even the medulloblastoma, which are also posterior fossa tumors, they occur in the cerebellum. Group 3 and 4 have either overexpression of EZH2, which is the enzyme that catalyzes the trimethylation of lysine 27, or truncating mutation that induce the loss of KDM6A, which is a lysine 27 demethylase. And really, another point that I would like to make, your hemisphere is probably a histone 3.3 problem, and your brain midline is probably a lysine 27 problem if you are a child or young adult with a glioma. And our initial discovery of histone mutation in glioma was furthered by other discoveries. So I spoke about the posterior fossa group A ependymoma, where E-Zhip and H3 and histone 3K27M coexist to specify this entity. We uncovered at the same time than the group of Guy Sauvageau in 7 to 10% of myelodysplastic syndrome K27M mutation in any of your histone 3 genes, or K27I, and some rare, super rare instances K27R. And we uncovered in about 15% of head and neck cancer that are HPV negative, mutation that lead to the disruption of the lysine 36 demethylation, including lysine 36M. The group of Adrian Flanagan and Peter Campbell in Oxford identified K36M mutation in chondroblastoma. 96% of chondroblastoma, which is a benign bone tumor, have K36M, and this is the only oncogenic driver. Similarly, they uncovered mutation that lead to G34W in histone 3.3, exceedingly rarely an arginine or a leucine in the giant cell tumor of the bone. And those mutations are the only oncogenic driver there. And it's funny because you never find G34W in the brain, but that's another story. And we uncovered in 20% of undifferentiated sarcoma lysine 36M. There is something that seems to appear here. Your K27 seems to specify mainly the brain and rarely AML or leukemias. And the K36, anything that affects K36M is more mesenchymal type tumors or epithelial tumor like the head and neck cancer that I spoke to you about. And the G34 is an in-between with a specificity for any of those residues. Quite intriguing. So why all of this fuss? Why those histone mutations are interesting? Why are we studying them? If you look at the sequence, the amino acid sequence, as I told you, it's amazingly conserved, even in plant. Arabidopsis thaliana has the same sequence, and this lysine or the glycine that are mutated are there. And if you mutate them, it's super likely that there is a problem that's going to arise. And what does histone 3 protein do? They're part of the nucleosome. The nucleosome is made of octamer of histones, dimers of histone 3, histone 4, twice. With histone 2A, histone 2B, you form this octamer and you enroll 147 base pair of DNA. And all of this leads to the compaction of your 2 meter 10 DNA into nothingness, into the nucleus. And the tail of those histones are quite important. Why? Because actually, they undergo post-translational modification. You have a writer that writes one post-translational modification node, something that reads it and act on it, and something that erases it, because some of them needs to be erased in order for whatever we need in terms of cellular processes during our development or during our daily life to be done. Gene expression is actually regulated. Our phenotype is regulated by the accessibility of our DNA and how the transcription is going to be made. And more interestingly, I told you about the canonical histone 3.1 and 2 and the non-canonical histone 3.3. They are not equal, even though they encode for histone 3 and they are part of the nucleosome. What you deposit on your chromosome matters, because if you deposit histone 3.1 and 2, you have your genetic code, those sequences, and then you have a histone 3 code. If you deposit histone 3.1 and 2, it's repressive. It's really one of those, it's almost like a space filler. Whereas the histone 3.3 marks active transcription, marks areas where you have your heterochromatin that you really want to silence, anything that's ERVs and stuff like this, and your telomeres. So histone 3.3 is also quite highly expressed during brain development. And probably this is one of the reasons why the mutations are more present in the brain than elsewhere, because there's something more specific that these mutations are doing there. And on top of that, you have your post-translational modification, a histone code that makes it actually very important what residue is mutated or not, because each one serves a purpose. Why is that? The way I view it is our genetic code is almost like your note of music. But remember that the same notes of music leads to the most sophisticated symphonies, songs, jazz, rap, whatever you want. It's just how you play with those notes of music with your genetic code that makes your genetic code meaningful. And actually the way I view it is that the epigenome and those histone, whether through the histone code or the post-translational modification, they give it a barcode and they make it sense when it needs to make sense. And unfortunately for those kids and the young adults that have those mutations, this barcode is completely wrong. Lysine 27 undergo monodi and trimethylation, and this is actually made by the PRC2 complex. EZH1 and 2 are the catalytic enzyme that deposit those marks. And this mark is a repressive mark, meaning it silences or compacts chromatin when it is present. Conversely, lysine 27 can undergo acetylation, and the acetylation of lysine 27 never coexists with its di or trimethylation. And the lysine 36, the lysine 27, when it's trimethylated, will actually recruit the PRC1 complex and repress and compact. So repression is K27 trimethylation. Gene activation is K27 acetylation. And the downstream lysine 36 get mono, di, and trimethylated by multiple different enzymes. I spoke to you about SETD2. This is the only lysine 36 trimethyltransferase. And the glycine 34 is just upstream, as I said, of this lysine 36. And the beauty is that lysine 36 trimethylation marks active chromatin. And really, what it does, it actually, you have H3K36 tri-reader that will go for transcription, regulation, and DNA mismatch repair. And those really are quite key. And the other thing is that whether you have a K27 trimethylation or a K36 trimethylation, they never coexist together. And even the lysine 36 dye creates a boundary. The dimethylation of lysine 36 creates a boundary with lysine 27 trimethylation. They cannot coexist together. They mark really boundaries. It's almost, this is your territory and this is mine. One does not go in the other. And remember, there's nothing that is void. All of your genome is marked by marks. And when one disappears, maybe others can take advantage of that. So how do those variants promote cancer? The first answer came through the beautiful work led by Peter Lewis, who was a postdoc in David Alice's lab, in the 2013 Science paper, where they exquisitely showed that the K27M or any K2M mutation on the lysine in the histone 3 tail actually acted as an inhibitor of the catalytic activity of the metal transferases that deposit the mark. And this leads to really, despite the fact that your mutant is barely 10 to 15% of the total histone 3 pool, it has a global effect, not only on the residue that's mutation, but across the board, because you're inhibiting the activity, the enzymatic activity of the metal transferase that deposit the mark. And so the first clue as to what those mutations would do and how they would do it came from a work which was co-led by me, David Alice, and Peter Lewis, where we work on the lysine 36M mutation that was uncovered in chondroblastoma in those undifferentiated sarcoma that I spoke to you about. Why? Because this was the only mutant there. This was the main oncogenic and only oncogenic driver. And we presumed it would be a little bit easier to model because we had a lot of problems modeling the K27M at that time. So what Xiaolu, who was a postdoc in David Alice's lab at that time, did is he overexpressed the K36M in mesenchymal stem cells. And what he exquisitely showed is that the moment you had overexpression of this, you inhibited any of the metal transferases, as what previously alluded to, but you inhibited any differentiation from a mesenchymal stem cell state to adipocyte, chondrocyte, osteoblast. You could not differentiate. And when we looked at the epigenome, when you had the K36M, this is a wild type. You see here the lysine 36 trimethylation, which mark the gene bodies, the lysine 36 dimethylation that work, it's like almost like a forest mark, vast area in the intergenome is there, and you have the K27ME3. And as I told you, those are like a strict boundary. The K27ME3 never coexists with the lysine 36 di or with the lysine 36 tri, and this is shown there in those wild-type mesenchymal stem cell. And the moment we introduce the K36M mutation, you lose, as expected, lysine 36 tri and lysine 36 di. And what happens is that this leads to redistribution to K27ME3, which spreads from its boundary to now paste the empty space left by the lysine 36 dye. Why is that interesting? Because if you remember from what I told you before, is that the lysine 27 tri acts in recruiting PRC1, and PRC1 is there to repress expression. So imagine you have your PRC1, and this is, we did your the PRC1 chips, CBX2 and Ring1B are two of the component of PRC1, the PRC1 complex. In the context of K36M, what we saw exquisitely was happening is that the K27ME3 was actually decreased in level. Remember, this is not a single cell, this is a bulk. So you have, it decreased in level. And this leads actually to decreased recruitment of PRC1. And actually PRC1 is relocated elsewhere. It's titrated, it's diluted. And when we knock down Ring1B, we could reproduce effect of K36M, which led us to say that in a normal context, you have K27ME3 that recruit PRC1, and your gene is off and your area in the genome is off. And they do not coexist with K36 dye that's deposited by the NSD1 or NSD2 complex. In the presence of K36M, you inhibit NSD1, NSD2, and SETD2, and you have a spread of the K27ME3, and you have so much more target for PRC1 that actually it gets diluted because everything is in rate limited quantity. You don't have enough PRC1 to keep the repression of those target sites that it has, and you need to repress now other sites. And so you have a dilution of the effect, a titration of PRC1 that actually leads to some gene being unduly repressed and other unduly expressed, and actually leads to this blockade and differentiation. So what about K27M? the first thing that was shown like exquisitely is that you had drastic loss. Immunohistochemistry could be very misleading. This is a wild type DIPG, and this is a K27M mutant DIPG. This is a K27ME3 marking. You presume that those cells that have K27M have actually no K27ME3 deposition, and the ones that have it are blood vessels actually to not carry the mutation. But no, there's still K27ME3 being deposited. And actually, this K27E, there is a conundrum. You have here like loss in areas, and you have areas where you have K27ME3 that's deposited. And this led to a lot of speculation from different groups. The group of Christian Helen thought that really it was really the high affinity site for PRC2 that could recruit it despite the presence of K27M. Other groups presumed, like the group of Ali Shilatifar, that the loss of K27ME3 led to increased K27 acetylation, because remember, K27 acetylation and K27ME3 do not coexist together, and once you lose K27ME3, the nucleosome could get K27 acetylation, and you had higher K27 acetylation in K27M tumor. And this increase in acetylation led to a novel oncogenic enhancer landscape and would repulse PRC2 from going there. Those are amazing hypotheses, but they weren't done in the context of isogenic models in the cell of origin where these mutations occurred, and work from my lab where we use isogenic cell lines in which we CRISPR the mutation, exquisitely showed in this and many other models. And in E-Zhip, as I will show you, that what happens is that PRC2 in the cell that acquired the mutation, the progenitor that acquired the mutation, is able to be recruited to its nucleation site. It starts depositing the mono, the tri, and the tri. But then, because it's catalytically impaired, because this mutation impairs the catalytic activity, doesn't have enough time to deposit mainly the di and tri. And actually, what we say is that it hits, PRC2 can hit the genome where it should land, but the spread of the K27ME3 and to lesser effect the K27ME2 is not happening. And actually it's one step down. In a wild type, you could get mono, di, and tri. In the K27M mutant, you get K27ME2 to the level of K27ME3, meaning really much less. And you barely get any K27ME3, which remains at the landing sites of PRC2. And other work in which we collaborated, and it's Peter Lewis who drove this work, shows that E-Zhip is actually EZH inhibiting protein, and I showed you how it has a K27M, does exactly the same. E-Zhip serves to show that you don't need the mutant to be incorporated into the nucleosome to act its effect. The moment it's there, it inhibits, and very small amount inhibit PRC2. And E-Zhip and K27M act to restrain the spread of ME2 and, more importantly, the K27ME3 to the nucleation sites. So we think that K27M poisons PRC2 at its nucleation site, because in embryonic stem cells, this is what you see. PRC2 is really only on CpG island, the nucleation site, and not yet moving. So why is that important? If you have a normal development in a progenitor, what it wants, it's actually to have stem cell gene and a lot of markers that are associated with patterning or with the differentiation to be silenced, because you don't need them. So you recruit PRC2, you put and you repress, you recruit PRC1 then, and you repress Hox genes that are associated with patterning, because once you have it, you don't want it to change, and genes that are associated with further differentiation, because it's not yet time to differentiate. And you have the expression of those stemness genes because they're important. And when you want to differentiate, then you keep the patterning at the area where you need it, but other area where you need differentiation, PRC2 is lost, you remove it, it spreads, and you express some. And PRC2 could go now and spread and silence those stemness genes because they're not needed anymore. What happened in K27M is that you get the first, you are a progenitor, you want to keep progenitor state, not differentiate, multiply and populate, and E-Zhip and K27M helps you do it indefinitely. There is no stop to that, because PRC2 is unable to move from its landing site and go and spread and mediate this needed differentiation, this needed silencing of stem cell gene, this needed in re-expression of genes that are associated with differentiation. And this is important because people have shown that throughout the differentiation in neuroprogenitors, but also in other cells, you have a wave of K27ME3 spread and of PRC2 movements that is associated with cell differentiation. And this is the one that's not happening. And this also explains why it's key when we put this mutation too early, it's lethal. When we put it too late, meaning in more differentiated or after the differentiation of those progenitors occur, it's not oncogenic. The only time where we could kind of have it is between E9.5 and E13.5 in mice. And this work was co-led with Paolo Salomoni, and Manav Petania did a lot of the experimental work in this. And Susie Baker's group also showed this exquisite temporal window where the oncogenic mechanism is possible. Which leads me to say that those mutations, E-Zhip or K27M and the K2M mutation, are what I call the Peter Pan syndrome. Peter Pan aged to a point and doesn't want to age further. And what those mutations are doing is restraining this progenitor from further differentiation. And it's cycling indefinitely, and it requires for further oncogenicity. And with time, all of the other mutation that makes it be detectable, because then it multiplies, it is there as a tumor. And this actually is shown by the work also of Mario Suva and Mariana Philbin, who described at the single cell level that the K27M mutation are oligodendroglial progenitor. So those mutations occur in this progenitor type and stalls its development. And the other mutations are there to either permit the expression or the cell to tolerate K27M or to increase its oncogenic potential. If that's true, if I remove the mutation, I should be able to see differentiation. And this is work that was done by Brian Krug in my lab, where he CRISPR'd out those mutations in those isogenic lines that I spoke to you about. And those are the lines with the K27M and the knockout in stem cell media. You're just asking the cell to grow as a progenitor in stem cell media. And this is when we push them to differentiation, either by FBS or by doing differentiation cocktails in them. And what you could exquisitely see is that the K27M containing cells are barely able to differentiate. Acquisition of GFAP with an astrocytic differentiation marker is really poor in here. And here in the K27M mutant, it was beautiful. More to that, the moment you remove the K27M in multiple different PDOX models, you increase tremendously the survival of the mice. So K27 is a needed oncogene at the beginning, at the mid, and probably at the end of any oncogenic process. And when we looked at those, this cell line, the BT245, and other ones, actually now we have data for the others as well, we ask ourselves, really, what happened to those tumor cells that were CRISPR'd, that are human cells? Well, they remain in the brain. We could detect human DNA in mice that were sacked about a year and a half after we had injected the cell lines and did never form tumor. They actually probably differentiated into astrocyte oligo, and we're looking now into what they were able to differentiate in the microenvironment of the brain and the pons where they arose from. The other thing, and remember I told you that when you have a mark that's lost, it gets replaced by another one. When K27ME2 and ME3 were not spreading, they were replaced in part by K27 acetylation in the intergenome. Usually K27 acetylation is really a very, there is not a lot of not a lot of nucleosome that K27 acetylation. It's really a low level mark because it marks promoters and enhancer, and it's an activating mark. In the presence of K27M, because they had so much more nucleosome, this K27 acetylation was there and was actually going in the intergenome. And mark my word, what it did over there was actually to activate the silent genome. And maybe this is a weakness in this cell, a therapeutic vulnerability, because if you push it further, either using DNA demethylating agent or increased acetylation, you could have the endogenous retroviruses that are normally silenced in a normal cell get expressed at a higher level and make this tumor a little bit more hot to the immune system. So just to summarize, what I told you is that K20 in a normal cell, you have K27ME3 that gets deposited at the nucleation sites of PRC2, and from there it spreads and goes to area which is needed to allow for further differentiation or silencing based on the cell's need. In the area we don't have K27ME2 and ME3, you have opposing mark like the lysine 36 di and tri or the lysine 27 that are there. And the K27ME3 really pays CGI, CPGI, and some CPG, and the intergenome where you have your endogenous retroviruses. In the presence of K27M, K27ME3 is really restricted to its bare necessities at the landing sites. And this allows for other marks to come and take over. And it's actually allowing for undue repression of specific genes that are involved in development and differentiation, and increased expression of genes that are more associated with stemness, and increase expression of the silent genome and endogenous retroviruses. So the main effect is stall differentiation, and maybe we could take advantage of the increased expression of the endogenous retroviruses. I want to target this tumor, I want to treat. So maybe by understanding the partnership, because the partners are, for now, what I can treat better. ACVR1 is a kinase, and really is key in the brain bonds, and it's clonal in the tumors where it is present. The same goes for FGFR1 or EGFR. So targeting those kinases is important. PGFR alpha is quite key for oligodendroglial signaling, and it's amplified in those midline tumors that are K27M. And it's funny because PGFR alpha amplification is mutually exclusive with FGFR and ACVR1. It's as if this is some oil that's needed as a kinase oil, that you get it with whatever partner you could find based on the stage of development or in the cell that acquired the mutation. And so those are the partners that I could target, and there are subclonal PI3 kinase mutations that get acquired with tumorigenicity that we could also target. The other thing is we could target the epigenome. Beautiful work done by Christian Helen, and I think this is real work. Shows that by inhibiting PRC2, you could potentially push the tumor further. So EZH2 inhibitor would remove the remaining K27E3, and because there is really limited K27E3 that stays in the K27M, you don't need much, and you wouldn't be affecting too much the normal cells that are nearby, and you could push those cells to die in there. Or the DNA demethylating agent or the increased acetylation in jointly that could help in the expression of endogenous retroviruses and maybe make the tumor more hot to the immune system, and then checkpoint inhibition could come. I think the future is in combination, taking advantage of targets that are unduly expressed in K27M, like the GD2, beautiful work by Crystal McCall and Michel Monge that uncovered this, and there's a CAR T cell trial, but also combining it with epigenetic drugs and kinase inhibitor, working from the inside and the outside. And I think this is a key to our future success for K27M and for E-Zhip mutant nucleome. So I spoke a lot about the K27M and a little of the K36M. How about the G34 mutation? As I told, they exquisitely occur in Histone 3.3 and work from Peter Lewis and other groups, and we collaborated with Peter on this, shows that really those mutations inhibit probably SETD2, which is a lysine 36 trimethyltransferase, and probably NSD2 and maybe NSD1 in depositing the di and trimethylation of lysine 36. And we showed in the giant cell tumor of the bone, for the lack of time, I won't go too much into that, that even this mutation that's barely 5%, the G34W is 5% of the giant cell tumor of the bone, acts slowly as a dominant effect and slowly reshape the epigenome and moves PRC1 and PRC2 from its target and dilutes it. And it actually, the effect in the end is to stall differentiation similarly. Okay, great. But what can I do? What about G34, R, and V in the brain tumors? And this is where I had the major chance of working with a group of Claudia Kleinman, who's really now my partner in crime. And Céline Gissa is a very talented graduate student in her lab. Jihad, Carol and Shreya in my lab worked also on specific aspect of this project. And the beauty is when you look at an elephant from too close, you're really mistaken. You don't see the full picture that it's an elephant. You could be mistaken. You could mistake it for a wall, for rope, for whatever you want. The image is self-telling. You really need to step back, and then you're maybe able to see the full picture. And this is what I think we did with this tumor. So the G34 mutant gliomas are hemispheric. They not find in the brain midline. They're really between 13 and 35 temporoparietal cortex, as I told you. And they're oftentimes mislabeled because primitive neuroectodermal tumor, 30% are actually G34R and V mutant, also ATRX and P53 mutant. They have dismal prognosis. And they have, for the pathologists, this dual feature that had led to a lot of debate, neuronal and glial feature, more neuronal, more glial. And when you look at the DNA methylation of those tumors, they all cluster, whatever you have by pathology, as one cluster by DNA methylation, which tells me it's the same molecular entity. Work started by Carol Chen in my lab, who told me, there are more mutation in PGFR alpha than you can imagine. And we went and accrued a lot of tumors from our collaborator and what was published in the website. And what we showed is that on 95 samples, where we had primary and recurrences, relapse pairs, whatever, is we have the G34 and V in associations with ATRX and P53, nothing new here, but we found 50% of PGFR alpha mutation. And to give you an example, in K27M, IDH, or wild type gliomas, you have barely any PGFR alpha mutation. There are 6 to 8%. And actually in the K27M, you have amplification mainly of PGFR alpha, which leads to its overexpression, but no mutation or no enrichment, significant enrichment in mutation. And 80% of the relapsed tumor in the context of G34RV had PGFR alpha mutation. And those mutations had something also that was quite unique in that they were mainly present in the extracellular domain of the kinase, not in the kinase domain as seen in solid tumor, other solid tumor than brain tumors. And this was alluded to initially for the rare mutation that she had uncovered by Susie Baker, this extracellular domain mutation in PGFR alpha that were most specific to brain tumors. Here, they were hugely enriched in the G34R and V tumor. And this is immunohistochemistry showing tumors that are wild type or mutant for the PGFR alpha, and how wild type and mutant PGFRA leads to increased signaling and to activation of downstream MAP kinase pathway, as shown here by surrogate phosphor-ERK staining. And when you acquired this mutation at recurrence, you had huge activation of this pathway. So from where those, I told you that K27M occurs in oligodendroglial progenitor. Can we pinpoint also the origin of those G34 mutations? And this is the beautiful work of Claudia and Celine, and we've been working together very hard because ever since we noticed this tempospatial distribution of the gliomas, but other embryonal tumors that we study, we worked in collaboration with Michael Taylor's group, who's mainly interested in ependymoma and medulloblastoma, and Maria Vladao is a graduate student in his lab, and we created an atlas of the developing brain at the single cell level, at the transcriptome single cell level. And this atlas was done by doing different time point at different location of the brain in the mouse, but also in human being, whatever tissue material we could acquire. And we did single cell transcriptoming and now single cell ATAC-seq on these. And we, on top of this, acquired every single data set that we could put our hands on that had done single cell analysis of those developmental time point and areas of the brain that we were interested in. And we created our own signature for progenitor and more differentiated cells that would allow us actually to say, this is a progenitor there, and this progenitor is able to differentiate. This is the pathways of differentiation. This is a speed. Those are the cycling cells. Those are the core transcription factor for this type of progenitor or more mature cells. And from there, to acquire bulk and single cell RNA-seq from our tumor and project them on that atlas and see what is it that they match best to. And came our first surprise. When Celine projected the bulk RNA-seq of G34 tumors, she saw that for glial tumor, it has 0 astrocytic or OPC signature. It has no excitatory neuron, post or prenatal, but it had an exquisite resemblance to prenatal interneuron progenitors and radioglial cells from the ventral forebrain. Not postnatal, prenatal, and not excitatory, inhibitory. And when you go and look at the brain development, your dorsal cortex will give rise to your excitatory neuron. Your ventral forebrain will give, through the ganglionic eminences and the lateral ganglionic eminences, rise to inhibitory interneuron. And the ventral radioglial cell that's here, that wants to commit either to an OPC in the LGE or the MGE, will have to choose its fate. And its fate will be dictated actually by transcription factor. And GSX2 and the DLXs, 1 and 2, are major transcription factor that, if acquired in this RGC, will specifically say to the cell to differentiate into interneuron progenitor and actively repress any differentiation in OPCs. And when we looked in the tumors, the G34 tumor, those transcription factors that specify interneuron progenitor were highly expressed, and they were not expressed in other glioma types. And further to that, when we looked at the epigenome, K27 acetylation, that marks these promoter and enhancer that are active, and K27ME3 that marks negative promoters, we saw that the DLXs and GX2 are really highly acetylated and actively transcribed. The size of the bubble shows the transcription levels. And so what we think is happening, and we had transcription data to kind of further show it, is that those tumors are interneuron progenitors that seems to be stuck in development because they upregulate genes that are seen in early interneuron and radioglial cell and actively downregulate genes that are seen in more differentiated interneuron, which lead us to think that this is when probably this mutation occur. And it probably, similar to K27M, stops further differentiation of interneuron progenitors. They could differentiate a little bit, but not as much as they would like to. How to reconcile PGFR alpha? I told you that PGFR alpha is highly expressed, it's mutated. And I'm telling you now with this schema that it's really only expressed in OPC. And I told you that GSX2 and DLXs, which specify interneurons, actively suppress OPC states. So how to reconcile both of them? And this is where chromatin architecture gives you the answer. And Jihad, Shreya, and Carol in my lab, in collaboration with Claudia and Celine, helped us find this. So we saw that this PGFR alpha gene is super expressed in G34 mutant gliomas compared to the other types. And the more mutated, the higher the expression it was. And when we looked at the promoter of PGFR alpha, we saw that it was actually situated, located on the genome where just downstream of GSX2, this transcription factor that I was telling you specify interneuron progenitor fate. And SHIC2 is the enhancer of GSX2 that drives the expression during development of this. And when we looked at the more highly ranked super enhancers in G34, SHIC2 and PGFR alpha promoter came as tops. And if you look, you acquire expression of this SHIC2, it's called here HS877, in the lateral ganglionic eminences. This is what drives expression of GSX2 and commitment to interneuron progenitor fate. And when we looked at the chromatin architecture of the G34 tumor, what we saw was happening is increased interaction between the promoter of PGFR alpha, and this is center, it's a 4C, virtual 4C that's center on the promoter of PGFR alpha, and this SHIC2 enhancer. And this is better shown here. This loops or super loop brings SHIC2 in front of GSX2 and allows the expression of GSX2 because you want it for interneuron progenitor. But in the case of G34 tumors, it's also allowing expression of PGFR alpha. And when you looked at the K27M and the IDH tumor, this loop was non-existent. You don't have it. It's really something that's specific to G34 tumor. So we asked ourselves, okay, it's present in G34 tumor. Is it present, this looping, is it present at all during neural development? Do you really need it to have GSX2 being expressed? And how the PGFR alpha enhanced interaction are they present also at early development? And we went and acquired data from Hi-C on embryonic stem cell. And because interneuron progenitors start at E13.5, we acquired data on ganglionic eminences, the area where those interneuron progenitor occur at that time point, but also from the cortex, because the cortex should not have interneuron progenitors. And what we could exquisitely see is that in a normal interneuron progenitor in ganglionic eminences, you had this increased interaction. But in contrast to G34 tumors, the PDFR-alpha promoter would be silenced by K27ME3. So interactions occur really in embryonic stem cells. And in embryonic stem cell, both GSX2 and PDFR-alpha are silenced by K27ME3 methylation. And in normal ganglionic eminences, because you want to acquire GSX2, K27ME3 is lost, and you allow for expression of GSX2, but you don't need PDFR-alpha, so it keeps being silenced by K27ME3. And this is what is lost, and we need to understand how it's lost in the context of the G34 tumor. The cortex doesn't need GSX2, so this loop doesn't exist in the cortex. And this is how, unfortunately, in G34 tumor, the co-option of PDFR-alpha takes advantage of a loop that's present during normal development. And by the change in the epigenome, the loss of the K27ME3 allows for the expression and probably subsequent mutation of PDFR-alpha. And when we looked at the single cell level at tumors and primary and recurrences pair, we could see that those G34 had a dual signature of neurons and astrocyte that's like signature, zero OPC signature or very limited OPC signature. And the moment you had a PDFR-alpha mutant, you had a higher astrocytic signature. And when we looked really, those cells co-express neuronal and astrocytic signature, and this is never seen in a normal progenitor in the fetal brain. You really either express one program or the other, which leads us to say that the G34 RNV occur in an interneuron progenitor that has a program of interneurons, and the co-option of the PDFR-alpha mutation and expression, initially expression, then mutation, leads to this astrocytic-like signature that gets acquired. And this is a sweeping event because the moment you acquire this mutation, it becomes clonal. And you need anything in tumors that have this mutation. This is the main clone that's present that expresses G34, ATRX, TP53, and PDFR-alpha mutation. And when we CRISPRed the G34 mutant in several cell lines, we saw no effect on oncogenicity, in contrast to the K27M. Whereas if you express the PDFR-alpha mutation in association with TP53, this was your major oncogene. Whereas the wild type PDFR-alpha and the G34 are really very poor oncogenes. And it's really what I think is that the G34 is only there to stop differentiation. And the only way this tumor could acquire oncogenicity is to find another pathway to have it kind of grow and express other things. And PDFR-alpha expression and mutation are a major pathway. So I hope I've convinced you that PDFR-alpha is quite important in the context of G34, and that G34, even though they're glial tumor, they're probably mislabeled because they are interneuron progenitors that are stuck in development. And this is important for modeling. And that PDFR-alpha co-opts the GSX2 enhancer for its aberrant expression, and this is targetable. With a word of caution, inhibitors of PDFR-alpha, even the new generation, do not act well on those extracellular mutant PDFR-alpha. And we need really to design new drugs that act on those mutations or already target the downstream signaling pathway, like the MAP kinase signaling pathway. And this summarizes my talk. I think oncohistone all lead to stall development in the progenitor they occur in. They stop its differentiation, they stop the lineage commitment, and they actually induce some shift shaping of the epigenome because the loss of some mark is replaced by other, which some may participate in oncogenicity, and others may actually be some therapeutic target that we could take advantage to. And actually, for me, the core is why is actually redistribution, titration of complexes that are associated with repression or with activation. In K27M, you really keep your restriction to specific area and you don't allow it to go to other places. In the context of G34 or K36M, you remove the repression from some area and acquire it in others that should never see it. And deciphering how to unlock those progenitor and what is the role of those added mutations that I spoke to you about may give us beautiful elements to treat those patients and to better understand their role in development. And this is beyond oncohistone, whether it's ATRTs, ETMRs, ependymoma, other things. This is a common theme. Whether it's a histone, which is the ultimate target, or its writer, erasers, whatever you readers that are mutated, they lead to similar effect. And this is beyond brain tumors and gliomas. It can go to other cancer like AML, like other things, IDH mutant gliomas. And this work could never have been done without the amazing collaborators that provide expertise, samples, advice, collaborator at the Dana-Farber at UCL, and now in Bonn for Paolo Salomoni, and other people across the world, and the people that participate in our iChange program, and of course our funding sources. And I hope that I will have a lot of questions to answer. Thank you very much for your attention.